Hello everyone, welcome to our video on human diseases and epidemiology, where we'll be exploring diseases with an emphasis on prevention and treatment in clinical practices, looking at specifically non-communicable diseases versus communicable diseases. We'll go through and define these terms with a special emphasis on infectious diseases. So from here we'll look at the chain of infection as well as the different modes of transmission involved in the epidemiologic triad pictured here. First up, let's define our non-communicable diseases. These are also known as our chronic and or degenerative conditions. They account for 71% deaths globally, with the top 4 non-communicable diseases that contribute to mortality being discussed below. Number 1 being cardiovascular diseases with 17.9 million deaths worldwide. Number 2 being cancers with 9 million deaths worldwide. Here we have breast cancer being the most prevalent cancer worldwide, whereas lung cancer is the most common cause for mortality worldwide. Number 3, we have respiratory diseases, things like asthma, COPD, with 3.9 million deaths worldwide. And lastly, number 4, we have diabetes with 1.6 million deaths annually, and the most of these are seen in the US. This is likely due to lifestyle factors. Risk factors specifically that contribute to this include hypertension and hyperdyslipidemia or high cholesterol. These all contribute to heart disease or cardiovascular diseases, and as such it is very important to follow clinical guidelines to help protect against the progression of these diseases. So these diseases are as opposed to our communicable diseases, which are defined as infectious diseases. Another term used here, often heard in epidemiology, is zoonotic, which refers to infectious disease that is transmissible under natural conditions from vertebrate animals to humans. And we have a few examples that I'll list below. Some long recognized zoonotic diseases include brucellosis from cows and pigs, anthrax from sheep, the plague from rodents, trichinolosis from swine, tularemia from rabbits, and rabies from bats, dogs, and raccoons, as well as West Nile encephalitis from birds. Backing up and looking at the overall big picture of infectious diseases, we do have an epidemiologic triad that is used to measure the control and or prevention of disease, which does require assessment of 3 components and their interactions. Number 1 being your agent, number 2 being your host, and number 3 being your environment. And we'll go through briefly and define these. And it is important to note here that your agent plus your host both exist within the same environment, and the time it takes for which an agent infects the host with a specific disease is what is most important to note here. This is known as your latency period, in which we can have a measurable response of exposure. And so backing up and defining the 3 different components of our epidemiologic triad, number 1, we have our agent, which is the infectious pathogen or microorganism that must be present in order for disease to occur. However, the presence of that agent alone is not always sufficient to cause disease. There are a variety of factors that influence whether exposure to an organism will result in disease. These include the organism's pathogenicity or its ability to cause disease, as well as the dose involved. From here, number 2, we have our host, and this refers to the human who can get the disease. There are a variety of factors intrinsic to the host, sometimes called risk factors, that can influence this exposure or susceptibility. Number 3, we have our environment, and these are the external factors that affect the agent. Lastly, we do have a fourth definition here that kind of bridges over to our zoonotic section. This is our reservoirs, which are the habitat of our agents. So for example, in the case of the bubonic plague, the Yersinia pestis bacteria is the causative agent that is carried by the reservoir, which are usually infecting rodents or rats and fleas that then bite humans who can become infected. This chain of infection must involve two pathways, one a portal of entry and two a portal of exit. From here, we have different types of modes of transmission that occur depending on the disease. Here, an infectious agent may be transmitted from its natural reservoir to a susceptible host in two different ways that can be further subcategorized, one being direct contact and two being indirect contact. With direct contact, this occurs through skin-to-skin contact, kissing, and/or sexual intercourse. Direct contact also refers to contact with soil or vegetation harboring infectious organisms. So, for example, hookworm is spread by direct contact with contaminated soil. And another subcategory under this direct contact is droplet spread. This refers to spray with relatively large, short-range aerosols produced by sneezing, coughing, or even talking. Droplet spread is classified as direct because transmission is by direct spray over a few feet before the droplets fall to the ground. Pertussis and meningococcal infection are examples of diseases transmitted from an infectious patient to a susceptible host by droplet spread. Whereas indirect transmission refers to the transfer of an infectious agent from a reservoir to a host by suspended air particles, this is known as airborne, inanimate objects, this is known as vehicle-borne, or animate intermediaries, this is known as vector-borne, with examples given within each category. Airborne being TB, vehicle being hepatitis by needle sticks, vector being malaria, and droplet spread being pneumonia. And with that said, our last section is going to cover clinical applications as they relate to the field of epidemiology. There's always a clinical approach versus an epidemiologic approach that can be taken to disease, with the differentiation here being the signs and symptoms of an individual versus the surveillance of an entire population. And with that said, I'm going to bring up an image below of the most common causes of infection seen clinically. These are organized by organ systems, and pharmacological recommendations to each of the microorganisms are available in the Public Health Epidemiology textbook in the description of this video. Going on to our black screen of spaced repetition to review important concepts within this video. Number 1, what is the time period between initial exposure and a measurable response of a disease? This is called your latency period. Number 2, malaria and hepatitis are examples of what kind of transmission? This is going to be indirect, with malaria being vector-borne, your mosquito being the vector, and hepatitis being vehicle-borne, your needle stick being the vehicle. Number 3, what types of approach are individual versus population-based? An example of individual would be your clinical approach, and an example of your population-based is your epidemiologic approach to disease. Lastly, number 4, what are the components of the epidemiologic triangle? 3 components here, number 1 being your host, number 2 being your agent, and number 3 being the environment of which both of these belong. And with that said, our last slide is an overview of everything that was discussed in the video. Please subscribe below, like, and share.